Well, thank you all for organizing this. I really wish that I could have been there in person. I just got back from traveling last week, and as a parent of two kids, that always makes things a little bit difficult. So, back-to-back travel doesn't happen these days. But I'm excited to be here. I don't often get to talk vocalizations anymore. My research has always had a translational focus, and it's taken me in different directions recently. But I really did want to spend the time and just talk about some of the work that we did, I think, really early on in vocalizations to discuss a little bit about the critical insights that having this model of affect or emotion has provided in substance use models. Kevin did a great job of setting this up. I honestly probably could have gone in either order with him and we would have talked about some similar things because we have a very similar background. But for those who don't know, a lot of the things we're trying to model in substance use disorder are perseverative drug seeking, escalation of drug intake, and a loss of control. But probably most important for USVs is the negative physiological states that coincide with the cessation of drug use. And as we're going to see, probably with more aspects of that, very similar to what Kevin had shown. And then susceptibility to relapse. So this all kind of comes together in one big cycle. And we like to look at every phase of this cycle because there's valuable information in each. Some of this involves preoccupation with the drug, the binge or intoxication phase, which is a lot of what Kevin talked about, and then of course withdrawal and negative affect. And we've had some insights into each of these phases over the years. And of course, the goal is to be able to stop somewhere after the withdrawal and get rid of some of these negative physiological states in order to prevent relapse. And so in some ways, as we're studying the brain, that's our goal. As Kevin noted, I think a lot of us think of this in terms of a model of an opponent process, right? And so the opponent process probably involves two different phases of preoccupation and thinking about the drug ahead of time, but also binge intoxication. And so, sorry, I pulled myself one slide off here for a second. With the main idea that initially drug use starts out with a peak in positive affect and eventually it subsides. And the theory, if you ask somebody like George Koob, who's been one of the main kind of proponents of this model in substance use, the main theory is that the negative affect tends to come during withdrawal. But I'm going to talk to you a little bit about both what we see in binge intoxication phases as well as the preoccupation phase that comes before that. And we see a lot of different places where I think negative mood comes into play. So self-administration comes in a very typical pattern. I'm going to be talking mostly today about stimulants, but you'll see there's a lot of similarities in what Kevin had shown, where we have a load-up phase that tends to be characterized by rapid infusions with a very short interinfusion interval, and then it's followed by a maintenance phase where animals will sort of regularly space their infusions. And we think of this as titrating their drug level. And if you notice that one feature of this is that when drug level falls, even just a little bit, you can see the rate of responding down below will increase. And so this is something that we think is really important for how animals choose to behave across a session of self-administration. And what we discovered is that if we experimentally manipulate this, where we hold drug levels either above satiety or below satiety, we can actually change the way that they behave to where we hold above their satiety threshold, then animals will stop responding altogether. And then if we hold it below, then we can increase their responding. So how do we better characterize the motivational states that perpetuate this? Because in terms of something like lever pressing, if an animal is pressing to avoid something negative or to seek something positive, it very much looks the same. And our answer to this was to use ultrasonic vocalizations. Kind of preaching to the choir here by discussing the different characteristics, but in rats, this is such a beautiful system because we have these negative affective vocalizations down in the 22 kilohertz range and a positive affect in the 50 kilohertz range. And so what we decided to do was to take animals and really manipulate drug level and use it as a variable to see how it changes affective states. So animals were all trained on an FR1 schedule with kind of a 40-second time in between their drug availability. And then we eventually switched this to a longer schedule where there's a variable interval and train them to regularly administer cocaine at 0.24 milligram per 200 microliter infusion. And then after this, we administered a series of drug clamps where during these clamps, animals were able to load up normally for the first about 90 minutes. And then after that, We either held drug levels below satiety using these kind of small micro infusions that you might think of like an IV drip, or holding their drug levels are pretty much right at their satiety threshold, self-determined by the rat each session, or well above it. And then we looked at vocalizations to see kind of what the affective states driving each of these would be. So what we found is actually very similar to what Kevin is seeing in the opioids here, which is that if we look before the session, there's a set of 22 kilohertz or negative affective vocalizations that happen just before the first infusion, and they subside as animals switch from being kind of sober to intoxicated over the beginning of the session. And then after that, we see a rise in 50 kilohertz vocalizations. But the really interesting thing for us was how transient this was. It's incredibly short-lived, and by the end of about 10 infusions, this goes away. So I think you can tell from seeing this in Kevin's talk with opioids. Here I'm looking at data from cocaine. We've also now seen this in bath salts like MDPV. This characteristic seems to be common across drugs, and regardless of their class, because now we're talking about at least 2, if not 3 different classes of drugs. The other really important thing is that this matches incredibly well with what we see in humans. So in some of the early work by Volkov, they would look at DAT occupancy, and this is where cocaine and other things bind, is at the dopamine transport for DAT. And what we see is that if you ask them about their ratings of high, and you can see that in the open circles, and then you look at DAT occupancy, which in a way is indirectly measuring drug in the brain, that the ratings of high subside far before the drug is out of the system. And in another study that came out around 1997, we can actually see that these, after an infusion, these positive kind of states like a high or a rush are more transient, and then they end up being followed pretty quickly by feelings of craving and low. And so this matches incredibly well with a lot of what we're seeing, and it proves that the translational relevance of this model is much better than we'd even originally thought. So then what happens if we manipulate drug level? Well, if we look at baseline vocalizations, we see a fairly low rate of both 22 and 50 kilohertz vocalizations if animals are just in the chamber. And interestingly enough, in these clamp sessions, if we hold animals either above their satiety threshold or at their satiety threshold, then we don't see very many vocalizations at all. But if we look just during the maintenance phase where animals are kind of titrating during the second week of self-administration, then we see an increase in negative affective vocalizations. And if we hold them below satiety threshold, then we see an even greater increase. And if we zoom in a little bit even more on that week 2, we find that when kind of that sawtooth pattern we've seen of drug self-administration, when the drug level falls to the lowest quartile, that's when we actually see the most of it. So not only is this predictive of low drug levels when there's subsatiety, it's also the fact that even if drug levels start to fall a little bit, we start to see negative affective states. And in some ways, what this is suggesting to us is that a lot of that maintenance phase is driven by negative reinforcement. Animals are continuing to respond in order to increase their drug level, and it's driven mostly by negative affect. They're trying to alleviate that negative state by taking more infusions. So another thing I was always interested in is how these responses changed over time. And this is data that we never published because I had this initial goal as part of my end phase doctoral work of reporting vocalizations alongside of single neurons in the nucleus accumbens. And we never really did get the two to line up very well. It's not, I think affect happens over very long time frames. That's not really that compatible with a lot of the neural signals that we like to look at, or it's at least hard to align them. But what we did do is, in what I was one of the earlier kind of vocalization detectors that we developed, was to take the data across these 24 self-administration sessions and model it. And so also consistent with the human data, if we model this, we just see a decline across about 3 weeks of self-administration that suggests that there's some major changes going on in the brain that fit very well with the opponent process model that Kevin and I both talked about. We see a decline over time. And just as maybe a little bit of a plug for some work that Kevin and I just did together, this matches really, really well with some of the long-term patterns we see in the way that neurons in the nucleus accumbens do change across many weeks of self-administration, and that there's kind of an initial lack of a correlation, and the more sensitive these accumbens neurons become to drug level, and the more this sort of negative correlation where more drug equals more inhibition of these neurons, then the more susceptibility we see in these particular animals in terms of their later escalation. So there seems to be some relationship where the limbic system is maybe showing tuning or changes to drug, and then across time we also see vocalizations declining. So there's some plasticity in the system that I think is something we should all care about. So and trying to kind of put all this together away from that aside, so we see a lot of different things. One is that the anticipation phase before animals start to self-administer drug is characterized by a mix of positive and negative affect. And then we see this transient bout of positive affect, followed by negative affect that's mostly driven by when drug levels start to fall. And then, of course, from a lot of experiments from others, we know that withdrawal is characterized predominantly by negative affective states. So this brings us to kind of the model of what we think is happening in most self-administration studies, which is that there are some very strong transitions where withdrawal carries into the moments before the next phase of self-administration. And again, we have positive affect as a key for just a few minutes. And then after that, most of the drug self-administration seems to be driven by negative affect and the avoidance of the feelings or even the start of feelings of withdrawal. And that starts as soon as animals even hit the maintenance phase of self-administration. So that's kind of a lot of little pieces of what we've done over the years, but I have a lot of people to thank for this as well. Some of the work that I showed at the end there of just the Cummins recordings, it's work by Ashley Crowley and Neeraj Sharma, both who were incredible undergraduates, and of course a lot of work in the West lab as well as the Wallace and Buschamp lab, where the bath salt data came from, and then the cocaine self-administration data came from. And as always, I have to thank a lot of really great funders, and I'd be happy to take any questions.
Thank you very much. I hope you can hear the applause from the room.
Yeah, I hear you at least, so we'll.
So you can stop share your screen, and then we will see you on the. Oh, great. So anyone who wants to ask some questions from the audience, please come here, because that will be much more and more audible. And we have a question from Zoom, and it's do you see animals which show normal drug intake patterns but do not vocalize? If so, what would that mean?
Do we see animals that have normal intake patterns but don't vocalize? This is a really good question. I think what I would say is that we see a range in the number of vocalizations. I don't know if I've ever seen one that doesn't vocalize, but we haven't taken the time to look at whether a low number of vocalizations versus a high number of vocalizations is predictive of some of the outcomes that we care about for self-administration studies. So that's a great question, but I don't think I have the answer to it, at least not any data that can speak to it.
All right, so we have another question. Do you know if the shape of the calls up versus downs is associated with a positive versus negative state?
This is another really, really good question. So if there's been a lot of work on this over time, and I think that one of the things is that I would say, and this is really others' work, is that the more complex the calls are, the more it seems to relate to some sort of heightened arousal. But I don't know if the up versus down is as important in my mind as just the overall complexity of the call. And I think this maybe speaks to a little bit of what Kevin had noted with the deep sweep side of things, where we don't have a perfect way of classifying or characterizing. So it's really hard to make comparisons. Is my sweep up the same as your sweep up and things like that? So it's really hard. We started out classifying things very simply into FF, FM, and trills, and then others moved to 14 classifiers or things like that. So I'm not certain we're making the comparisons fairly yet, even this many years later. So.
Okay, great. In fact, Zoom just exploded with questions, and a question from Piotr Popik. Piotr, Gifty from Warsaw. And so what about other reinforcers like water, sucrose?
I guess you could take this in a lot of different directions, but do they behave the same way? I think several have seen positive affective vocalizations for sucrose, but I don't think that they behave in quite the same way, even though I do think that under the right conditions, for example, there's some really incredible work out of Bart Vogel's lab years ago saying that if you give enough sucrose and then you give naloxone, you can actually get a withdrawal from sucrose. So I think a lot of these things could behave in the same way. But I don't. Depending on what comparison you're asking about, I mean, feel free to clarify, but I think it's some similarities, some differences, I guess, if I simplify the answer.
All right, so another question, I think that would be the last one. If you look at the peak frequency of calls over time, is there any indication that there is a gradual decrease in frequency before a transition to 22s, or does it seems to be a discrete jump?
This is a great question. I believe that we've looked at this over time, and I don't know that I can say that there's any clear pattern, at least not that we've seen. Maybe there are others who have. I don't know if others have seen anything specific, but I don't think that I've seen that in the data we have.
Mm-hmm.